Hi, my name is Sue Ludman, and I'm a senior research associate at the Allen Institute for Cell Science in Seattle, Washington. And today I'm going to give an introduction to microscopy and biology. In particular, I'm going to give a lesson on viewing cells in 3 dimensions, or 3D, using the Allen Institute for Cell Science 3D Cell Viewer. First, I would like to describe a common tool that all biologists use. It's called a microscope. What is a microscope? It's a tool that lets us look at things that are small. On the left, you can see a light microscope, and on the right is what's called a confocal microscope. Light microscopes use regular light bulb as a light source. Confocal microscopes use lasers tuned to a precise wavelength as the light source. Using a confocal microscope with lasers allow for fluorescent dyes, which can be red, green, yellow, blue, many different colors, to be illuminated and visualized. Also, cells that are imaged with a confocal microscope are optically sectioned in the Z dimension, and these slices can then be compiled into what's called a Z volume, allowing for the whole volume of the cell to be looked at. You can look at the whole compiled volume of the cell, or you can actually look at specific slices within the cell, and that allows for one to examine specific structures much more carefully. Different microscopes can zoom into different levels of detail, and more detail can be observed with a confocal microscope. If one was to look down a light microscope, you might see something like this, depending on your sample. What's shown here is pollen. The other classic example that I like is some pond water. You just take a little bit of pond water, put it on a slide, look under the microscope, and you can see little critters swimming around, maybe some algae, other particulates that you might find in a pond. On the right is a beautiful image of a tumor cell. And this image was acquired using a confocal microscope. You can see here that there is much more detail that can be observed using a confocal microscope. In red is the cell membrane. In blue is the DNA, or basically it's the nucleus, which is where the DNA and RNA and all the genetic material resides. And in green is one of many, 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 many structures within a cell. This green structure in this example is called microtubules. And you can see that it has different sort of fine detail compared to the other two structures. Next, I'd like to talk about the general concept of a cell, which many of you have probably already learned about in some of your biology classes. And I'd like to use an analogy of a car here. Just like a car has many parts, cells also have different parts, each of which have a specific and very precise function that when all together allow the cell to perform what it needs to do, whether it be a brain cell, skin cell, whatever type of cell you might think about. Shown here is just some of the classic structures that you can find in cells. In pink is the nucleus in this case. Again, this is where the DNA, RNA, and genetic material resides. And there's like this little pink membrane around the nucleus called the nuclear membrane. In the light gray is the cytoplasm of the cell. And inside the cytoplasm of the cell are many, many, many structures, a few of which are shown here. The Golgi apparatus and endoplasmic reticulum are involved in protein processing and many other functions. The mitochondria gives the cell the energy that it needs to do many, many functions such as divide, grow, and also power for other things that occur within the cell. And then the other example is the microtubules shown in green. Microtubules are part of the cytoskeleton of the cell, so it gives the cell structure. It also can repurpose itself, and it's involved in the division of the cells as cells grow, and it separates chromosomes. And all of these organelles within the cytoplasm are contained by what's called the plasma membrane, and that is shown in the darker gray here. The next concept I'd like to talk about is fluorescence, because my confocal microscopy uses fluorescent light, as I mentioned before. Fluorescence microscopy lets us use light, again, a specific type of light called fluorescent light, to look at specific cell parts. Again, let's use the analogy of a car here. On the left is a VW bug, a yellow VW bug driving down the road that we're all used to seeing. But as we all know, a car is made up of many, many, many different parts, right? The lights, the steering wheel, the gas tank, everything under the hood of the engine. They all have specific roles, and each of these parts come together as a whole to allow the car to do what it needs to do. Fluorescence microscopy basically makes a single part of the cell glow. This would be like making a single part in the car engine glow, which is shown on the right. If you open up a hood of the car, one of the many, many things under the hood of the car is a spark plug. And so what I'm showing here is the spark plug with a little bit of a fluorescence green tag. That's sort of my analogy here. So having this fluorescence allows the structure to light up and let us see it better. But we can only see one part of the car at a time. We can only see one part of the cell at a time by using this approach. We need to use fluorescence, again, one at a time in each structure. Sometimes we can put two together, and then we try to figure out how all these different, if we look at each one independently, and then we bring them all together, we can understand the whole of the car or the whole of the cell. So earlier I showed you a cartoon image of a cell, but it was a flat 2D image of the cell. But cells actually are not flat. They have a volume like a ball. And confocal microscopy, again, uses lasers to optically section through this volume. So that's what this slide is showing here. So we use the microscope to acquire single slices that we can assemble into the 3D volume. On the right, S1 and S2 are showing the different slices. If you can imagine, you might have 5, 10, 100 of these. And when you put them all together, you will get the volume that is in the middle there. So thinking about the different parts of the cell that I just described, if you look at the red ball, that might be like the nucleus. Green could be a different structure, blue could be mitochondria, etc. So the individual optical sections, like S1 and S2, are they're collected one at a time, and then they can be brought together, summed together to create the whole volume of the cell. And there are benefits, again, to seeing the whole volume, just to have a sense of how the whole cell looks. But if you're interested in specific structures within the cell, you might want to take a few of these slices and look very closely and specifically at those structures. In confocal microscopy, 3D imaging using confocal microscopy is a good way to do this. Machine learning scientists, computer scientists, can make models of cells to learn more about how these structures function by compiling very large data sets of many, many, many cell volumes. So if we're looking at a cell with one structure and then looking at another cell with a different structure and making many, 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 many images like this, they can take all this data and feed it into a model and create some sort of sense of what these structures are doing, and using artificial intelligence can sort of predict, based on their models, what the cell might actually do or what it had done before. Shortly, we're going to be doing a demonstration of using one of the Allen Institute for Cell Sciences online free tools called the 3D Cell Viewer. And I want to just, before we go there, reiterate the idea that we are going to tag one specific structure with a fluorescent tag. In our case, we're using a green fluorescent protein. And the two examples that we'll be using in the demo is many cells using mitochondria. Again, I talked about that earlier. It's the powerhouse of the cell. It provides energy, allows the cell to grow and do its functions, grow, divide, and many other things that it needs to provide the cell. It's like the energy for the cell. So mitochondria is sort of a general term, and there are different proteins within the mitochondria. In our case, we've tagged a protein called TOM20. So that's what you'll see in the demo. The other structure we're going to look at in detail is the microtubules. Again, a microtubule is a general term, and in our institute, we have taken a specific protein called alpha tubulin and tagged it with, again, a green fluorescent protein so we can follow it. And microtubules, again, are very beautiful structures that are involved in actually the structure of the cell. It's like a scaffolding, almost like in a building. But it also can be repurposed when the cell divides and grows and multiplies. Microtubules are also involved in pulling chromosomes apart and allowing the cell to divide. Images can also have channels or colors. It's a concept I want to introduce. We've seen this image earlier. And as we go through the demo and your work in this exercise, when you're looking at cells, you just want to ask yourself, what do you see in each of these images? What do each of these colors represent in this image? How do they look differently as far as, is it a smooth texture? Is it uniform? Are there spots where the color resides and not? Because that's where it's telling you where the protein, where the organelle of the cell is located. And how might we use these different channels to see more information within a single microscope image? So we'll work through some examples where we're looking at some of these channels, some of these colors, one at a time, and then we will combine them, and how the power of being able to combine the different channels gives you much more information about the cell. Okay, now we're going to do an activity where we're going to use the 3D cell images that we talked about, and we're going to use the Allen Institute for Cell Science 3D Cell Viewer tool, and I'm going to demo that for you now. When we open up the viewer, we're going to look at some 3D cell data. We're also going to look at different organelles. We're going to take the image and we're going to connect the images that we look at to the fluorescent microscopy techniques that we use to create them. And in so doing, we'll be able to describe where organelles are located and learn about the different distribution of organelles within the cell. You can follow along in the student handout for instructions and questions about what we're doing here and the cells themselves. Okay, the first step we're going to do here is to navigate to the Allen Institute for Cell Science 3D Cell Viewer tool. You can use the link here. So just put this into your browser. And once you do that, you will end up on this page at the Allen Institute for Cell Science. You can see there's a lot of tools on the top, but we're focused here on the 3D Cell Viewer. It takes you to the page actually of all the images that we have currently in this tool, which is 31,000, almost 32,000 different cell images. But the first thing we'd like to do is move from this images page, as you can see here, and kind of come over here to the right where it says viewer, and then click on the viewer. I would just take a minute here again to explain the goal. We're going to explore single channels of a single cell and explore the cell in the Z dimension. Again, to remind you, cells are not flat. You can have a flat 2D cell image like I've shown you before of these cancer cells. We have an X and Y dimension. If you put a slide onto a microscope, you have X and you have Y. And then you're going to have the laser coming in the Z dimension, which I'm showing here in the teal where it says Z. You can see how the cell is actually a volume. So when I talk about X, Y, and Z, X, Y is on the stage of the microscope, and we're using the lasers to cut through the cell optically in the Z dimension. Okay, So the first thing we're going to do here is to select a mitochondria structure or cell containing where the structure has been tagged with the green fluorescent protein to TOM20, which is a protein within the mitochondria. I've highlighted a few of the steps here, and I'm going to demo this now. Okay, so here we are again on the image page. The first thing you want to do is select the image cell type. When you highlight this bar here, image type, you can either look at the whole field of view of cells or you can look at a single cell. For this first step, we're going to look just at one single cell. So we select single cell, and then we're going to look up the protein tag. And in this case, we're going to choose TOM20. You can pick any cell here that you want to use for this exercise. But I have found one that I like. So I'm going to go ahead, and you can remember, the tool will remember which image you have if you note it. So I'm going to put in the name of this particular cell, which is AICS-11 underscore 30 underscore 4. Again, we can use any of these cells, but this is the one I would like to use today. So once you have your cell up, you can double click on that cell, and it will be put into the viewer here. And you can see it's kind of cool as it starts moving around. You can go ahead and sort of square it up a little bit and get it to stop moving, which is sort of nice for these demo purposes. And then on the right over here where it says tools, go ahead and deselect the membrane, deselect the DNA. You can also change the color that you want to be visualized here. I like to look at things in black and white initially. So you can go ahead and select for the TOM20 to be shown in white. At the bottom here, you can see the X and Y dimensions, which we're not going to mess with today, but they're also shown in these bars up here. You can see how those change as you change these different sliders. But for demo purposes here, I'm going to go ahead and reduce down the number of Z slices. And you can see what that's doing to the image above as I do it. Then I'm going to go ahead and move this Z. You'll see how you're going through the cell. And you say, oh, that's not very bright. I'd like to make it a bit brighter. So we can go down to a viewing editor, scroll all the way down here, and there are a couple of adjustments that can be made to sort of help you have a better view of the image that you have here. There's different rendering qualities. We can go ahead and use max. Then you can also change the intensity level, and you can see how that's changing the image above. I like it to be pretty crispy, so I would turn it up to about here. Then you can change the brightness of the structure from to like not even being shown to like being over saturated. Something in here looks pretty good to me. And then you can also play with this density. Okay. I also want to point out this tool over here. You can see my pointer again. This allows you to zoom in and out. So let's go ahead and make this a little bit bigger so we can see it. Again, you can turn the image around. And you can go up and through the image like this. I've already done this part, but this is just for the handout. You can see as you move the slider up and down, how the structure of the mitochondria, depending on where you are in the volume of that cell, how it changes. So what are some of the observations that you can make here? Can you describe what you're seeing in the cell? And what parts of the cell can you see? What can you not see? How does the structure change as you move through Z? Observe the shape and position of the structure and the intensity of the image. Based on the image of a living cell, how would you describe how mitochondria look? Observe closely and be as detailed as possible in your description. Next, as I talked about earlier, we're going to explore the power of confocal microscopic 3D volume images. So again, on the left, we have a flat 2D image of a cell. Remember, we can acquire different slices and compile it into a volume. And then these volumes can be rendered using computer models and computer imaging techniques so that machine learning scientists can create models to determine how different structures work with each other and on their own and their functions in the whole cell. Okay. Okay, so next we're going to select a different cell, a different organelle, a different cell structure. We're going to look at microtubules now. And again, microtubules is a general term for this cytoskeletal protein, but we've labeled this cell using a fluorescent tag, and we put it onto a protein within the microtubule called alpha tubulin. And we're going to go through the same steps, and then we're going to compile different channels on top of each other. So I'd like to demo that now. Okay, so instead of the mitochondria here, again, we're going to go up to image cell type. We're going to select a single cell. Okay, and then in the protein tag button, go ahead and hit alpha tubulin. Again, you're going to get a huge collection of different cells showing alpha tubulin tagged with GFP or green fluorescent protein. Again, I have my cell that I would like to use for this demo, AICS-12_30_8. It comes up with this nice, beautiful cell. Go ahead and double click on this cell and it pulls it into the viewer. Okay. Just like we did before, we're going to play with some of the tools on the right here to kind of optimize this image here. So let me do that. Again, we have black and white. And first I'm going to get it to stop moving. Just click on it, it'll stop moving. We're deselecting. So normally you might have these things pop up. Go ahead and deselect DNA, deselect the membrane. Have only the alpha tubulin channel selected. Scrolling down to the editor, again, we can look at the rendering quality. I like to use max, get the best, sharpest image we can make. Go ahead and play with some of these other tools. You can see what they do, kind of blows it out. It's very, very bright, more bright than the mitochondria. So go ahead and bring that down a little bit. And you can also adjust how much you want to have around with the density of the structure. And then go ahead and bring this up so that you can see this beautiful cell. You can tighten down the Z section here. So as I was saying before, when it's totally wide open like that, you're looking at the entire volume of the cell. So let me actually go back so you can see that. So you go here. So this is all the Z slices. Here's our balloon that is a cell. And then you can click on it and you can move it all around and see that it actually is a volume, which is pretty cool. And then we'll hold it right here for a second. Now we'll go ahead and tighten down the Z. You can go all the way to 1. I like to have like 2 or 3 in the so-called 2 or 3 slices in the stack. So that's this little slider here and here. If I put them on top of each other, it'd be just 1 slice. But here's about 3 slices. And then you can go up and down through the cell. And oh, that doesn't look bright enough for me. I'd like that to be a little brighter. I'd like that to be a little brighter. You can see more structure. There you go. You can see how that's moving in the X and Y dimension, how it's changing these lines as I turn the cell. Again, you can zoom up and down and see the beautiful, that's kind of a nice view right there of the detail of the microtubules. Okay. Okay, as we did for mitochondria, we did for the microtubules, you know, tighten down the Z slice, optimize the brightness and intensity that you want, and then go ahead and zoom up and down through Z to see the different details of the microtubules. So now, what are your observations? What can you see by looking at this cell image? What parts of the cell can you see again? What can you not see? If you compare the cell to the cell that we looked at before with mitochondria, is there anything that's sort of similar? What's different? How does the structure change as you move through the Z dimension? Observe the shape and the position of the structure and the image intensity. Okay, so now we're getting into the concept of the channels. We're going to add the membrane channel to the microtubule channel. And so in order to do this, what we're going to do is go back to the demo. And in this case now, go ahead and select this membrane channel. For sort of optimize, I like to optimize each channel independently, and then you can overlay them on top of each other. So here it's showing up as pink. Let's go ahead and make it red since we use a red dye in our data. Okay, then come down like you did before. And as you can see, what you cannot see, but what I know is if you look into the middle of the cell, you get a very nice outline of the membrane. But if you sort of imagine you're going to the top of the cell, it's a ball, remember? There's signal on the top of the cell, you're moving down through the ball to the bottom half of the cell, and things look different whether you're on the top or the bottom. But let's just optimize to the middle. So we'll put the Z slices here in the middle and go ahead and adjust the intensity. There you go, adjust some brightness and the density. Maybe like something that's a little heavy like this. And that's too much for me. Bring it down a little bit. Okay. Again, as I just showed, you can move up and down through Z. So this is a different cell, but you can see again what I was trying to show in the demo. On the left is the top of the cell, and it's not a perfect ball with a perfect membrane. It kind of juts out and moves around depending on the pushing and pulling of the cells around it. So you can see these protrusions in some cells, not all cells. And then as you move into the middle, like I did, you can start to resolve very like a line almost. And then as you move through, things change as you get through the cell. Okay, so now we have our membrane channel, how we like it. And we had our tubulin as we adjusted it. So if you click on both of these now on the right, it will overlay the two channels on top of each other. So what I like to do now is go ahead and make the tubulin channel green. So if you saw what I did here, click on this box. It gives you this array of, you can make it any color you want, but I know we've tagged our alpha tubulin with a green fluorescent protein. So for me making sense of things, I like to have it in green. I actually like this really bright green. Okay. Now it seems a bit noisy. Doesn't mean you can't go ahead and optimize some of your settings. So you can go down again and change things. I would change them one at a time, actually. So I think red is really quite bright here. So bring that down a little bit. There, sorry, it'll be here. Once you have that one set, you can click that off, and then you can put on the alpha tubulin. And just so we can see it a little bit more clearly, I'm going to bring that signal up a little bit. And you might have to go back and forth a few times. Okay. So here we are with these two channels. Again, as you start at the top of the cell, you see the membrane, and you can see some of the green microtubules coming through here. You slowly move through the Z dimension. You're moving through the cell. You can see how the amount of microtubules, depending on where you're at in the cell, how it changes, how bright or not bright it is, and how much of it is actually present there. So now I've moved here. I think that's too noisy. So you might have to go back and forth to try to make yourself a nice image. See the detail you want to see. Maybe even zoom up. Put the red one back on. So there you go. Top of the cell, through the cell, bottom of the cell, and the two different signals can be visualized. That's just what I'm showing here to highlight both channels. We turn them both on, optimize them, like I say, as you like, and then move up and down through Z. So what are your observations here? What can you observe about this? So when you have two channels on, you have a labeled structure, which is tubulin, and then you're putting a membrane channel on there. When you only have, versus what we did earlier, when you only have one channel on time. Having two distinct parts of the cell being shown at the same time gives you a lot more information. Can you describe the orientation and shape of the alpha tubulin, the DNA, and the membrane in relation to each other? Now I mentioned DNA, but we haven't quite turned on the DNA yet, have we? So that's what we're going to do next. So here we go. We'll go back to our image. I'm going to turn it down a little bit. I'm going to turn off the membrane, turn off the tubulin, and I'm going to turn on the DNA. I don't like this really light blue. I like it a darker blue. So there we go. It's a little bit, stands out much better for me. Go ahead and optimize this signal as you like. You can have it like this, maybe play with the different tools here. Maybe something like this, okay? And we've already sort of optimized our other two channels, our membrane. Now you can see the nucleus and the membrane. And now you can see all three structures. If you take off the membrane, now you're only seeing the nucleus in blue and the tubulin in green. Let's just look at some of these, just two of these together with the nucleus. So at first we'll just kind of scroll through the cell in the Z dimension. We're on the bottom of the cell. Look at all the microtubules there. You get in the middle, there's the nucleus, and then you get to the top, you have another population of microtubules on the top of the cell. Now, if we take off tubulin and look only at the cell membrane, so at the top, again, we're getting into the middle of the cell. There's the nucleus, and then there's some structure of the membrane on the bottom of the cell. Move to the middle and put all three channels on. we scrolled through the nuclear channel only, and hopefully we can make some observations of what occurred there. Then we overlaid all 3 channels. So now that we were able to overlay 3 different channels, what can you observe about this cell once you do have all 3 channels turned on? So you're going to have the labeled structure, the membrane, and the DNA all together, versus when you only have one structure, or you only have the 2 pairs at a time. Now you have 3 structures, and so you have a lot more information there. Given all of this information, what are you still curious about related to this particular cell, to this cell image, or maybe the way this image was taken? Okay, the last thing I would like to do is just remind you that you can give yourself an added challenge, and you can repeat any of these sorts of back and forth tools with not only just looking at a single cell, but when you take an image, the way our cells grow, they're right next to each other. So that's called a field of view, or a field of images. And you can go ahead, instead of selecting a single cell here, you can select a full field. So if you'd like to explore further, you can do that. And again, all you would do here is go ahead, instead of selecting single cell, select full view. And I'll just take one of these, pop it in so you can see what that looks like. And you'd have to go through all the optimization of each of the channels again, like we just did, and to make your pretty picture, and then scroll up and down through Z and see what you see. Okay, so that concludes this lesson and demo of the Allen Institute for Cell Science 3D Cell Viewer. I would just really like to thank the entire cell science, and beyond the cell science, the whole Allen Institute. We work as a team at the Allen Institute, and there are many, many of us that work together. And of course, special thanks to Paul Allen, who's the Institute founder, for his vision, encouragement, and support. Thank you.